to draw some conclusions about genetic linkage, to estimate genetic linkage. So this is based on a simple logic, which is that the closer two genes are on a chromosome, the less likely it is that they're going to be separated by crossing over, right? Let's consider a chromosome here. And here is one gene that codes for one trait. And here is another gene that codes for another trait. There's a lot of distance between those genes, so it's more likely that a crossing over event can occur in any one of those sections of DNA. If, however, those genes were closer to each other, let's say here, they're really close, then there's a much shorter distance over which the recombination event can take place, so it's going to be more rare. Geneticists were able to utilize that fact, that logic, before they were able to sequence the genome at all, and they used the observed ratios of non-recombinants to recombinants to estimate how close genes are in a chromosome. This is before they had the ability to sequence anything, so they didn't know. They knew about some genes, but they didn't know where exactly on the chromosomes they were. So they had to use this very indirect method by making crosses and observing. What were they observing? The number of recombinant offspring, so the ones with different combinations of allele than the parents, and the number of total offspring as a proportion of the number of total offspring. This is just for EdXLB note. For the rest of you, like, you can know that, you know, the further the genes are, the more likely they are to get separated by crossing over, but don't worry too much about the actual calculations of the recombination frequency. That's an example. So a student was investigating the inheritance of two characteristics in fruit flies. One is body color, gray is dominant to black. The other is wing length. Normal is dominant to vestigial. Vestigial means like tiny, basically. After performing a dihybrid cross with two homozygous parents, he then interbred their offspring. This produced the offspring ratio shown in the table here. Yeah, I'm going to come back to this table, so don't worry. Using the offspring ratio and provided experimental information, state how the body color and wing size genes interact, and explain how you reach this conclusion for 5 marks. Let's break down the phrasing of this question. What is our context? Our context, obviously, like all of this is also context, isn't it? So let me just circle it here in green. All of this is also context. And further, yeah, sorry, context is previous page. What is my command? My command is actually 2 command words: state and explain. What is my direction? My direction is that what kind of like genotypes am I going to talk about? So how the body color and wing size genes interact. Sorry. Take a minute to structure this question in your mind. So I'm only focusing on how they interact, right? There's no need to go and construct a Punnett square here. Don't feel that you have to do that. Let's look at the answer together and break down the different marks here. So the body color, first of all, we're going to explain that the body color and wing size genes exhibit autosomal linkage. That is one, like our first kind of conclusion. Why? How do we know this? What is the explain bit now? The F2 generation phenotypic ratio is roughly 3:1. Whereas if the genes were unlinked, we would expect a 9:3:1 ratio, like with any other dihybrid inheritance. Homozygous parents have these genotypes, and so the only possible gametes that they can produce are these. And is this gamete from this parent, this gamete from this parent. Therefore, all offspring would both be heterozygous for both traits. The 3:1 ratio that is seen in the F2 offspring, the F2 generation is basically the second generation when you have crossed the offspring of one generation. As the majority of gametes formed are homozygous for the dominant and recessive traits, as these alleles are inherited together on the same chromosome. A small proportion of recombinants are seen due to crossing over, and which leads to the formation of these recombinant gametes. Therefore, some organisms will have these genotypes here, which is what we observe in the table.